So, no more waiting. So, good evening, everyone. This is Dr. Mahesh Veluswamy here, and thank you, everyone, and thank you, Dr. Arthi, for joining with us today. So, today we'll be taking introduction on molecular modeling, and it will be presented by Dr. Arthi Venkatesan, and she is graduate and also she did PhD in computational biology, and now she is working as a scientific specialist in Discovery Studio. So before passing it on the slides to Dr. Arthi, I would like to give a small notes for the participants. So during the meeting, kindly switch off your webcam for better buffering, and also you will be always in mute mode. So if you want to ask any questions, you could drop your questions either via chat box or you could use raise hand option. And always use your original name for any kind of discussion. Like if you want to ask chat or via oral, use your original name. Then last but not least, the most important request that try your best not to leave the meeting before it ends. It might give big motivation to the tutor. So let's start with the lecture. So Dr. Arthi, I'm going to make you as a presenter and you can start your lecture now.
Yeah. Good evening, one and all. I'm Arthi. So I'm currently working as a scientific specialist in Altum Technologies. Probably you might have
Arthi, sorry to interrupt. Can you share your slide?
Yeah, I have already shared, no?
No.
No. Once again. One second.
Yeah.
Is it visible now?
Yes.
Yeah. Hi. He has already given introduction about me. And currently I'm working as a Discovery Studio software. Probably you might have heard about the term Accelerate Discovery Studio. So I'm a technical head for my team. So here I'm going to discuss about homology modeling. You might have heard the term homology modeling, I guess. If you know anyone about this homology modeling, kindly put it in chat box so that I can understand how many of them know about homology modeling and everything. So can I wait for 1 minute or 2 minutes?
Yeah. So guys,
to know the answer.
So you can let us know if you know about molecular modeling before. If you have previous experience, just let us know how many of you have experience in molecular modeling.
So fine, even if you doesn't know the answer, let me explain you on the upcoming slides. So homology modeling is nothing but to determine the protein structure. Okay, so for example, you might have studied about central dogma of the cell, right? So how the protein structure sequence is determined. So in case if you have a protein sequence, so how you're going to give it as a 3D structure model. That's why here we are going to discuss about this in detail. Okay.
Okay. So Arthi, can you just make it simple words? What are you going to address in this lecture? Just in points.
In this lecture, we are going to discuss about how the protein structure determination can be done and what are all the X-ray crystallography methods, what are all the methods are available to predict the protein structure and how you are going to deposit the available structure or predicted structure in RCSB protein data bank and what are all the prediction methods are available, what is homology modeling, what are all the steps involved in homology modeling and what is the how to do validation. Okay.
So Arthi, maybe we can say like this: you are going to give them an answer. What is homology modeling and how we computational bioinformatics going to help with predicting the structure?
Exactly.
Right. Okay.
Exactly. Yeah.
Okay.
So, for example, most of the I think most of the molecular biologists or biotechnologists will be here. So, to resolve the functional conformation of the protein structure is a biggest challenge in molecular biology, right? So to analyze the functional knowledge of the protein is an imperative knowledge. And also the greatest challenge for a structural biologist to predict the structure of the protein. Okay. Here, what are all the different methods are available to determine a structure? Gel filtration or Western blotting and side-directed mutagenesis. So here you can check the quantity of the proteins and everything. But in to study or to design a protein, to evaluate the protein, to model the protein, you can go for atomic or electron level methods. That is called X-ray crystallography, nuclear magnetic resonance, that is called NMR, and cryo-electron microscopy. Hope you people have studied this in detail. I'm just going to discuss what are all the basic concepts are available in that, and how as a computational biologist will design a protein structure. Okay. So coming to X-ray crystallographic method, so there are some steps involved in X-ray crystallographic method determination, right? So for example, you will be purifying the protein, you will be crystallize the protein, you will be collected the data. Once you have collected the data, you will go for structure solution, that is called phasing. So after you determine the structure, you will go for structure determination, that is called model building and model refinement. Okay. So here you can check the electron density map also. So as soon as if you get the electron density map, you can go for 3D structure model. Okay. In case if there is any problem with the crystallization, then again you redo the crystallization process. In case if you are getting the problem in phasing, then again you need to start from the crystallization, that is called purifying your protein. Okay.
Okay,
So this is the
Arti, here you are
yeah
trying to say that already existing are physical methods
yeah yeah
that
yeah
available for crystallizing. Okay,
crystallization, yeah,
okay,
exactly.
okay.
So and what are all the advantages and disadvantages associated with X-ray crystallographic and NMR methods. Okay, so for example, for example, the best example is you can grow the crystals of the protein and high resolution microscopy, and here you can calculate and refine the model, and based on electron density maps you will get the good model, and protein function can be determined, and RMSD, you know very well, it is called root mean square deviation. And what is the disadvantages associated with this type of methods? Okay, it will take weeks to years, as I have mentioned earlier in my previous slide. Okay, if there is a phase problem or crystallization problem, then again you need to redo it from starting. Okay, it will take time, right? So not every protein can be crystallized. In case if you are going for a membrane protein, it is very difficult task to get the protein structure, and it is very time consuming as well as expensive method. And the size limitation, there will be some particular limitation in the sequence length also. Again, various resolution, for example, you can determine a high resolution and low resolution. That is the biggest task in biggest disadvantages associated with X-ray crystallography and NMR methods. So as a biotechnologist or as a microbiologist or molecular biologist, you might have done a protein crystallization, or you might have studied, or you might have done crystallization technique, and you have designed a protein. Okay, so once you have designed the protein, what you will do, how it will become a 3D structure, and probably you might have seen three-dimensional structure of the protein, I guess. uh, Dr. Magesh, they know the 3D proteins, right?
Yeah, I guess they know what is 3D structure
Since you
or
people,
XYZ coordinates.
yeah, since you people have already done molecular docking and all, they might be knowing about the 3D coordinates and everything.
Okay,
Yeah,
yeah,
okay,
yeah,
I got the message. Yeah,
yeah.
yeah. So you got the 3D structure and you got a three-dimensional structure of your protein. Then you are going to deposit that in your RCSB data bank. Okay. So what are all the steps involved in that? Okay. For example, you have that sequence and you have a protein structure itself. So first thing is you need to check the sequence. Uh, so that is a major task. So to check the sequence, you have blast P and blast N, which is also available in RCSB page itself. Okay, if you go to RCSB, I have given the link in reference. Okay, if you go to that link, you can you will be getting the clarity how to deposit a protein structure. Okay, here have just given the few hints. Okay, So, and the second point is check the ligands. So, in case you have crystallized the ligand along with your protein structure, you need to check whether the structure is already available or it is a new structure or you have done any modification in that. So, for that, you can go for ligand expo. And there is a chemical component dictionary which is also available in the PDB page itself. Okay, and the third thing is to prepare the data for deposition using PDB, extract, and SF tool. There are two tools are available to deposit the structure. The one is called PDB extract, and another one is SF tool. So there are tools like I have given the tool links also. Okay, so if you go to this site and if you go to RCSB site itself, you can find this, right? And next thing is to validate the structure. So once you have done deposition, then you need to check for validation. You have already done through electron density map and everything, and again in RCSB they will be checking the structure. Okay, if it is not good, they will be rejecting the structure. Okay,
So,
that is the major
Arthi,
concept.
I would like to
Yeah,
emphasize one point here. People
yeah.
might not know what is RCSB, so it would be nice if you tell them it's kind of database, or can you,
Yeah.
yeah, yeah, just tell them what
Yeah,
is RCSB.
yeah. RCSB is nothing but protein data bank. Magesh, is it possible to visualize the data bank?
No, no, it's okay, it's okay.
Okay. I didn't give it here. Okay. Probably next lecture, or I'll send you the link for RCSB. Okay, so that will be better for you to understand. Uh, in RCSB data bank, all the protein structures will be deposited. So whatever previously, I think before 2011, so not 2011, 2001, I guess, even if you model a protein structure, you can deposit your protein in RCSP. Okay, now the option is not available. Am I right, Dr. Magesh?
Yeah.
Yeah.
So it's a type of database that
Yeah.
the structures are deposited.
Yeah, exactly. So for example, previous cases, some students, what they have done is they have modeled the structure and they have deposited. Like that, they have found so many errors. Then they have framed a basic protocol how to deposit a structure and how to what are all the structures can be deposited, so that you can go for X-ray crystallography and NMR. Even if you are making it in advanced level, like if you are using Discovery Studio or Schrodinger software, then you can submit your structure. For example, if you are making a structure for COVID, you can deposit. There are some rules and regulations. There are steps involved in that. I have mentioned about the steps involved in structure deposition. Yeah, okay. So in case if you are using NMR structure, you can deposit the structure using edit and edit NMR. I have given what are all the steps involved in that. For example, if you go to edit workstation sftools.rcsb.org, you can find it out, and what are all the steps involved to submit your NMR structure. Okay, so next is protein structure prediction method. So far I have discussed about wet lab techniques and how to deposit a protein structure. Now here we are going to discuss about protein structure prediction methods. So what is structure prediction? So as a bioinformatician, as a computational biologist, what you're going to do? Okay, for example, I have say for example, you can take COVID-19, right? Okay, if you're taking a COVID-19, you're finding out a vaccine. So if you're going to find out a vaccine, you need to know the protein structure, right? So if you need to know the protein structure, you have tried with X-ray crystallography, NMR, and it's not possible in this case to make it as simple. Then you can go for this computational biology methods. So for example, homology modeling for proteins with 3D similar structure. Okay, and for threading and ab initio method, there are steps involved in that. So for example, for homology modeling, the sequence identity should be uh, greater than 35 percentage. Okay, and for threading, you can go for less than 10 percentage, or greater than 35 percentage also can be used for threading method. And ab initio method, it is like starting from a scratch. You doesn't know anything about the structure, you cannot even find out the similarity between the structure, then you can go for ab initio method. Okay,
Okay, maybe
so
Arti, here
yeah.
I would like to summarize them, like uh,
Yeah.
some points. Okay, so
Mm-hmm.
I guess that there are some biotechnology students, so
Mm-hmm.
they might wondering how the
Mm-hmm.
biological sequence are converted into computer, and how we bioinformaticians contribute in identifying the vaccines. So the answer
Yeah,
will be
exactly.
placed here. So the answer will be, you might heard a lot of news like there are the sequence discovered for COVID-19, but at the end of the day, how they are going to use that sequence to find the vaccine. So there the bioinformatics make a big role. For example, so far Dr. Arthi explained what are all the classical methods that used for crystallizing the structure, but also she mentioned what are the disadvantages associated with it. The one disadvantage is it time consuming. So what we bioinformaticians normally do, we take the sequence and we convert them into structure using different bioinformatics tools and methods that we'll be talking later. So in that specifically, we have three different part. Like if the given query sequence, let's say COVID-19, and you want to predict a structure for the COVID-19. So first we go with homology modeling. If the sequence length is 30 percentage or more than that. If the sequence is less than 30 percentage, we'll go threading. If the sequence length is it's less than 10 percentage, in that case we'll go for ab initio. It starts from scratch. So this is the overall idea, like how the structural mathematics going to work in the real example. So
Yeah.
I guess this
Uh,
might give you a good idea for all the students. So maybe Aarti, you can start from here. You
Yeah,
can,
yeah, yeah.
yeah.
As he has already mentioned about the techniques and method, and he summarized it, right? So next we are going to see what is homology modeling. Okay. So for example, homology modeling. So first we should know the term for homologous proteins, right? So what is homologous protein? I have two protein structure, and I need to, for example, I have a human protein, and I need to check for a another protein. Okay. So I will check it through monkeys samples, monkey sequence, right? So that is called conserved region can be predicted through two proteins structure, and you can find out the similarity between that. So highly similar protein are also called as homologous protein. And in homology modeling, we can call it as a comparative modeling also. What is comparative modeling? We'll be having this question, right? So comparative modeling is nothing but you are comparing your sequence with another similar protein, another homologous protein. Okay. So, and the next thing is to predict the protein structure based on the template. So here template is nothing but you are going to check for a monkey sequence. That is the template, right? And the next thing is analysis of protein function. So you're going to analyze the protein function. If you doesn't know the protein sequence or a protein structure function itself, you can find out through homology modeling. Okay. And even rational designs of the protein with increased stability and novel functions can be determined. And 30% of the sequence, as Magesh has already mentioned, 30% sequence identity is required to build a model. And if you're not getting 30% sequence identity, or if you're not getting enough sequence identity, then you need to go for ab initio method. You don't have any other option. You need to go for that. And in case if you are getting more than 30% or more than 50 or 60 percentage, then you can go for threading also. Okay. And even the evolution structure is more stable, and the changes occurred at slower rate when compared to its sequence. Here I have mentioned about distantly related sequence. Say for example, human and mice. Okay, they are distantly related sequences, and we have the reason why we are using, as a biotechnologist, you probably know about why we are using mice, right? And in case if you are having 40% or 60% homology or similarity, you will be guaranteed to adapt a similar structure. Just like a crystallography structure, you will be getting a similar structure with a good model. Okay.
And Aarti, I have a question
Yeah.
here. Maybe the same question people will also have.
Yeah.
Is the comparative modeling and the homology modeling terms are similar?
Yeah.
Okay.
Homology modeling, we will call it as comparative modeling also.
Okay. So it's it's just like
The
you're
reason,
calling.
the reason why we are saying the term comparative modeling, we are looking for a homologous sequence. We are looking for a template sequence. Okay,
Okay.
that is the
Okay.
reason we are mentioning here as a comparative modeling.
Okay. And also one more doubt. What do you
Yeah,
mean
yeah.
by template?
Template.
Yeah.
For example, here I will be explaining it in my next slide.
Okay. So maybe here to give a just clue, template is the similar structure of
Template is
the given
Yeah,
target
similar. That
sequence.
is called homologous sequence,
Okay.
homologous structure. Okay,
Okay.
for example, you are taking one protein as a human and another protein as a mouse.
Okay.
Okay, so your human protein is your target protein and your mouse protein is a template protein. That is your homologous protein. Okay, the
Okay.
template protein is also called as homologous protein. Okay.
Okay. Okay. And also one more term that maybe you can explain. What do you mean by distantly related sequence? It's as a
Yeah, yeah, I have mentioned.
yeah.
Yeah, for that I have mentioned already. In case if you're taking human and if you're taking mice sequence,
Mm.
which is similar. Human sequence is similar to monkey sequence or mice sequence. Mice is distantly related, right? That's why I have mentioned distantly related sequence also you can find out.
Okay. So
Not only, yeah,
it you mean like evolutionary
identical.
related
Yeah,
evolutionary related.
evolutionary related sequences.
Okay. 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 Thank
Yeah,
you. Yeah.
okay.
Yeah.
So the next thing is how you're going to build a model, how you're going to align the model, how you're going to build a model. That is the biggest task. So we know very well, we have gone through the steps for X-ray crystallography method and how to deposit the structure and how we will build a model, how we will build a homology sequence. Okay. So for example, I have a target sequence. I have already mentioned about, for example, here we can take it as a human. Okay, human has a target sequence. I'm going to look for a monkey sequence, right? So for monkey, I can say template identification and selection. So I'm just going to look for another sequence that is called template. Okay, so I'm just going to select my template based on the homologous sequences. Okay, then I can go for alignment. For alignment step itself, I can go for structural alignment and multiple sequence alignment. Structural alignment is nothing but you're going to see helix and loops. Okay, so probably you might have studied about central dogma of the cell. In central dogma of the cells, after that you might have studied about protein structure. Okay, protein structure is made up of primary structure that is called linear chain of amino acid and your secondary structure and your tertiary structure, and your secondary structure contain alpha helix and beta plated sheets, right? So here you are going to see the structural alignment through that based on your alpha helix as well as beta plated sheet. And the next thing is multiple sequence alignment. You're going to align with multiple sequence and then you're going to find your template. Okay, it's not like you're going to find only the monkey sequence. You're going to find out many sequence and based on that you are going to say this is the identical sequence. Okay, and after that you're going to build a model. Okay, once I have done alignment, I used to build a model, right? Then only I can get the protein structure. Okay, so once I have built the model, I'll be refining the model. So once I have refined the model, I have to go for evaluation. So in case if the model is not good, okay, I need to redo the steps from starting. Okay, and in case if it is if the model is good, you can go for validation. Okay,
Okay.
validation we can go for, we can use Ramachandran plot.
So,
Got it?
Arthi, maybe I
Mm-hmm.
can add some more terms here, like to
Yeah,
make it
sure.
simplify. So, you meant for a given target sequence, we are going to search similar sequence, but maybe you can say like this: for a
Mm-hmm.
given target sequence, we are going to search templates against the PDB database. So, for example, let's say you have human sequence, or you are going to find the templates. So, it's not going to be a non-redundant sequence database. It's going to
Mm-hmm.
be against structured data, PDB structured database.
Yeah, you are
Target
right only, but
sequence.
the thing is,
Mm,
yeah,
yeah,
the reason why I didn't mention about RCSB here, it differs
it's
from
it's it,
software to
yeah,
software. Yeah,
it's not about RCSB. In BLAST
yeah.
itself,
Any,
we have a different database. So one is non-redundant database.
Non-redundant
We have
database.
only sequence. They don't associate with any structural details. Whereas in structural database, like protein, again, like you can also say template database, they always deposit sequence ID, sequence associated with the known structures. So you should emphasize that we need to search the query sequence against the template structure database. Yeah, that's what I want to convey.
Okay. So hope you understand what are all the steps involved in homology modeling. So the next thing is I can go for I'm explaining everything in detail. Okay. So what is sequence alignment? Sequence alignment is nothing but you have a target and you have template sequence. So you are going to see the similarity between target and the template sequence. Okay. So why you are doing this sequence alignment is to get the model accuracy. Okay. Then only you will be getting the better model accuracy. Okay. So, for example, query sequence is 90 to 90% identical of the template. The model will be identical like a crystal structure. In case I have already mentioned about X-ray crystallography and NMR spectroscopy, right? So here, crystal structure is nothing but if you are determining from X-ray crystallography or NMR or cryoconservation method, so that is called crystal structure. Okay. So if your query sequence is above 90 or 99%, it's a well and good model. Okay. And in case if it is around 50 percentage to 89 percentage, we can say it's a reliable one. It's a highly reliable one. You can take this, you can consider this also as a model. Okay. If you're getting 30 to 49% identity, you need to redo it. Okay. So you may have find errors in the loop. And in case if it is less than 30% identity, it is not at all a good model. Okay.
Okay.
So here I have
Arthi, as here maybe some confusion might arise. The three terms: query sequence, target sequence, and the template. So here the query sequence and the target sequence are both are same term, right?
Yeah, exactly.
Okay. And the template is the similar sequence that we are going to search against query sequence or target sequence. Yeah.
Target sequence.
Okay.
Yeah,
Okay.
exactly.
Good.
Guys, is there any doubt with that template or target so that we can rectify it here itself?
So if you guys have
You can comment it
some
in
confusion
you
with
can
the
comment it
terms,
in
you
your
can
box.
tell us. Yeah.
Yeah.
Okay. I guess no doubt. Can move.
Yeah. Okay. Okay, so the next thing is alignment correction. So first thing I have done sequence alignment. The next thing is alignment correction. How I am going to align it. Okay, so for example, it is a little difficult to align two sequences. Okay, if the similarity or if the percentage sequence similarity is low, it's very difficult to align it. Okay, so for example, simple sequence alignment which has highest score based on your matching or pairing of the residues on the two sequence. Here in the left image I have mentioned about template and model as well as both good and bad model. So for example, if the template with the good model, that is okay, first thing is bad model, right? The model which has less gaps, okay, those things are considered as a it's not a good model. You can redo the sequence alignment and then you can go for alignment correction again. And in case if you are getting template with a good model, which means there are some gap, but it is aligned conserve. For example, cysteine, arginine. So they are conserved, right? So like that, it should be conserved with the other amino acids. Conserved, I have mentioned about if two homologous protein is similar, that is called conserved model. Okay. And in the right side image I have mentioned about cluster W alignment. In case if you are not getting proper alignment correction, you can take your sequence, you can take your target sequence as well as template sequence, and then you can go for cluster W website. There is a server called cluster W. Here I have mentioned cluster W. Just go to cluster W and then you can paste your sequences and you can do if you search it, then you will be getting your alignment like this. Okay, it will automatically find the gaps alignment and mismatches are available. So for example, this one is Q9, the first sequence is your human sequence, and another one is the target. The first is your target sequence and the second one is your template sequence. So you can compare your target sequence with your template sequence. So those are amino acid residues. Okay, in case if it is similar, you will be getting asterisk symbol. Okay, in case if it is not similar, you'll be getting double dotted. If it is if it is little similar, you can go, sorry, if there is any gap, you will be getting like this. Okay, so the underscore which represent gap.
Okay.
Any doubts in that?
So maybe I
You
can
can
ask some
clarify it
question
here.
here,
Yeah.
Arthi. So
Sure.
you're talking about alignment correction. Yes, of course you can do that, but how much it is going to be reliable?
Reliability based on, for example, if I don't do alignment correction, if I have find more gaps in a protein sequence, I'm just taking that sequence and modeling it. Okay, so
Okay.
what is the use of doing that? Okay, it's of no use. You will not be getting a good model, right? So that's why you are doing alignment
But
correction
how
to
much
rectify the issues.
reliable or the
You're asking about the percentage or
like how much confident we can keep it for further step? And is that
in case if it is not reliable, then you can redo the alignment and then you can rectify the error, then you will get a good model. Then it will be reliable and precise, right?
yeah, for the redo the
Yeah.
alignment, do we need to have any manual edit or everything will be done by only cluster W?
You can do manual edit also, no, through some PyMol or any other
Yeah.
softwares.
Okay,
Yeah.
but
Even Mega you can use for alignment correction and everything.
so you are trying to say that until when the sequence alignment is corrected, we can keep it in the loop and do the correction until we get proper alignment.
Exactly.
Okay.
Yeah, if you don't do the alignment correction, it's of no use. Again, even if you are getting a model, it's not a good model. It's not a precise model.
Okay.
Okay, it's just like you have done homology modeling with the sequence. That's it.
Okay, so and also I want to emphasize the symbols. So the star, it's I mean, I would just want to recall the star is for conserved region and semicolon for identical residues. Or what do you mean by it's not
semicolon is not identical. Semicolon is not identical. It's a mismatch sequence.
Okay, and
the asterisk is a match sequence, and underscore is a gap. It is not matched with any other sequences.
Okay, and there are also one more symbol. You can see
dot symbol.
the plus symbol might come in between any sequence. So you have any idea what the plus symbol implies in the sequence?
I will verify it and will let you know. Or if you know the answer, you can tell them, bro.
So guys, if you are wondering what is the plus symbol implies in the sequence, so that is simply nothing but it's an identical sequence. So you might come across the plus symbol in the later sequence alignment.
Yeah, even in the asterisk symbol, it's also a matched sequence, identical sequences.
Okay. Yeah. Thank you.
Okay. The next one is backbone generation. I have done alignment, sequence alignment and alignment correction. Then I need to look for backbone generation. What is backbone generation here? You might have studied about alpha helix and beta plated sheets, right? So here a backbone is nothing but your alpha helix and beta plated sheets. Okay. So to generate the backbone coordinates from the template for the aligned region. So for example, you have already aligned it with the template and you need to check for the backbone coordinates. For example, here you have some, for example, the first one, okay, human as well as 5y3t. Here you will be finding in AEEM. Okay, here you need to find out the if it is if two are conserved. Okay, here the if you find helix or loop or helix or beta sheets are available, you need to find out the backbone coordinates there. Okay, if most of the aligned sequence are identical, backbone 2 will be identical. If two sequence are identical, similar, if you're getting like this, for example, MDE till L, it is same, right? So this means the backbone also identical. Okay, if the template that generates a backbone with least error is chosen. So in case here I have already mentioned, I'm not going to look for only one template. I'm not going to look for only monkey sequence. Okay, I'm going to look for many other sequences. From that, I will be choosing the best template. Here they have mentioned why we are using a best template. Okay, to rectify the error, in case if you're finding more error in the backbone, you will not consider that particular template. Okay, and backbone of secondary structural elements also can be considered, and that those things can also be maintained. So variation can be seen in loops and turns. Okay, so further it is a loop modeling. Loop modeling is nothing but you're going to model a loop, so you need to know what is loop, right? Loop is nothing but it's a region of your query sequence which is not aligned of the template. If it is not aligned with the template, that model is as considered as a loop. Okay, so in loop modeling, it is like knowledge based method. PDB is search for normal loops. PDB, I have mentioned about RCSB PDB. We are going to search with that RCSB. And energy based method, you will be looking for long chains as or built by sampling Ramachandran conformations and everything. And for insertion, you can go for coordinate of missing residues is given in the loop conformation. Okay, in case if any missing residues are available, you can go for loop conformation. You will be inserting a particular amino acid. For example, here you can. So here, in the last one, there is no. I think you can find so many gaps, right? So here, if you insert any amino acid related to that, that is called insertion of a loop. Okay, that's what I have mentioned here. If you have any doubts in this, you can
So,
ask or you can.
Arthi, in the previous
Yeah,
slide, can you come back to the previous slide?
yeah, sure.
The next one. So,
Yeah,
from this slide, you're just trying to say that these backbone generation and loop
backbone.
modeling
Yeah,
are some
more
protocol
important.
used in structural refinement, and backbone
Yeah,
generation deals with secondary structure refinement,
yeah,
whereas loop modeling deals with the
loop.
refining the gap in the alignment
Yeah,
sequence,
missing
right?
residues. Yeah,
Okay,
missing
okay,
residues. Yeah.
okay.
Guys, any doubts with this? If you have any doubts, we can clear it here itself.
I guess no,
I guess no doubts.
it's a direct answer. Yeah.
Yeah, yeah. Okay. So the next is side chain modeling. Okay. So first thing you have done sequence alignment and alignment correction, and then backbone generation, loop modeling. Then you're going for side chain modeling. So side chain modeling is nothing but if any side chains are missing, then you are going to model the side chains. Okay, based on your template or generated model. Okay, so I have mentioned placing side chain coordinates. Okay, you're placing your based on your template model, you are placing your side chain. Okay, and side chain remains more flexible on the surface and tends to adapt more multiple conformation. And if you find any wrong backbone, the effect of the side chain building process can be affected. Okay, and then prediction accuracy can be highly dependent based on your so that you will be getting a good and refined model. Okay, and what is model refinement and
Arthi,
structural yeah.
in the side chain modeling, uh,
Mm-hmm.
how it differs from the previous backbone generation based on the
Yeah,
purpose. How it
backbone. Yeah, backbone we have seen here. I have mentioned, no, one second. Backbone here we have mentioned about helix and beta sheets, right?
is a
But
secondary
here
structure.
you're,
Here you
secondary
are,
structure.
okay, and here
Here
completely you're
you're
dealing
going
with the individual residues. Okay,
individual residues in case if the side chains residues are
with
missing.
related to
Okay,
their flexibility, right?
flexibility. Yeah,
Yeah,
exactly. Okay.
in that case, not all the
Mm.
amino acids are same in terms of flexibility. For example, the residues proline, histidine, all those has closed ring.
Yeah.
So how you are
Yeah.
going to deal with those type of residues when it comes to side chain modeling? Are you going to keep the flexibility for the proline residue like completely? Because if you keep the complete
No, depends upon the amino acid residues also, no? So I will mention what are all the residues left for side chain modeling also.
okay,
Okay.
and what are the
After
tools?
this class,
Yeah,
yeah.
yeah, and what are the basic tools that you will for side chain model?
Yeah, yeah, I'll come to that later.
Okay,
Okay.
okay, okay. Thank you.
Okay. Then model optimization and structure refinement can be done through in the same homology modeling process only. And model optimization, you can go for real in vivo confirmation. If you have done through in vitro and in vivo methods, you'll be getting the same model if you follow this type of steps. Okay. In case if you're not, if you're missing some steps, okay, if you build a model, it is not a good model. You will not get a real model. Okay. And further, if you align the model, if you're getting the model as a good if you consider it as a precise model, then you need to go for molecular dynamic simulation to study in depth about the atomic coordinates. Okay. And motion of the protein, we can check through molecular dynamic simulation. And there are n number of tools and servers are available for all the three methods. For say for example homology modeling, we can use this model, and there are servers also available. iTaser and Robusto also are available for homology modeling. Even for commercial softwares, you can go for a Discovery Studio and Schrodinger. Apart from that, we can Magesh, do you know any commercial software
You can
apart
use,
from?
yeah,
Yeah,
you can use the Schrodinger, as you said, and Discourse Studio.
yeah.
And
Apart from
one more,
no, I'm
there are
yeah.
one more tool that I know. Maybe we can ask directly from the students if anybody knows other than these two, Discovery Studio and Schrodinger.
Yeah. Otherwise, I will send you
Okay.
in detail about this. Okay.
Yeah.
And ab initio method, you can go for CATS fold server, and also you can use Modeler. Modeler is the first modeling software which has been discovered for homology modeling. Okay.
Okay.
Not only homology modeling, all the three modeling can be done through Modeler software. Okay.
Okay. And
And
here in
thread.
Abinishio, maybe you can say about AlphaFold,
Yeah, yeah.
the
Also,
recent trend. The AlphaFold completely used machine learning to predict the fold. So it's a trending
yeah.
one. Yeah.
Yeah.
Abinishio.
And threading, you can use iTaser, Lomax, Robusta, and there are some other tools also available. If you need any other tools and servers, I
Yeah.
will.
Okay.
Uh,
So other
I'll send you once
famous,
the.
so other famous tool would be you can use Pirate 2 and also
Yeah,
Rosetta.
yeah, Pirate.
You should not forget mention the Roberta or Rosetta. Okay.
Yeah,
Yeah.
yeah, sure. Okay, so we have done all these steps. So finally, we have to go for validation. So for validation, we have to look for RMSD. I have already mentioned about X-ray crystallography method. Then you can find easily about RMSD, right? So here you will be validating through that root mean square deviation. So you know very well the concept of RMSD to calculate average distance between two superimposed proteins. So here two superimposed proteins, which means your target sequence as well as your template sequence. Okay, so you will be superimposing is nothing but you are merging two proteins. Okay, so I don't know whether the merge term is correct or not here.
Comparing,
Yeah, yeah, yeah,
just
comparing.
comparing. Yeah.
Exactly, since we are in bioinformatics field, we used to say this term superimposed. Okay, we're not saying it's merged, right? Okay, so with the help of superimposing two protein sequences, that is two protein structures, that is your target sequence as well as your template sequence, then you can find out the average distance between both. Okay, so D stands for distance, and remaining things I have mentioned. And if you want to obtain a good model, the structure RMSD will be 1.5 Armstrong, and if even if it is 0.0 to zero point 0.5 Armstrong, it is essential identical. Okay. Otherwise, probably they have read about RCSB, or you might have uh, given lecture about databases, right?
Not yet.
RCSB data. Okay, so I'll give a next class about
Okay.
RCSB database. Okay,
Okay. Okay.
so there you can find out the angstrom. I mean, your angstrom and 1.5 angstrom, or which means it's a good fit or good model. The quality can be determined through RMSG. Okay, and then Ramachandran plot. So probably you people have studied basic biochemistry or advanced biochemistry, right? There you have studied about Ramachandran plot. Here you have your protein sequence and you have built a model through those steps, and you have your model with you. Then you need to look for Ramachandran plot using Rampage server. There is a server called Rampage, and with the help of Rampage server you can identify the errors in that. Okay, so you can check the quality of the model. Okay, so distribution of torsion angles in the model can be determined through Ramachandran plot, and residues with pi and psi angles sterically disallowed conformations can be studied through that. And here I have mentioned about allowed most allowed region, disallowed region. Okay, most allowed region which represent red color, and which means there is no steric clashes are available in that. Okay, disallowed region which is nothing but atom closer than the van der Waals radii. Okay, and in case if it is allowed region, that is called LO1, with residues have atom with interatomic distance. Okay, you can check for the interatomic regions and distance, and then with your van der Waals radii, and you can conclude the quality of the model. Okay,
Hello, Arthi.
so
Hello,
yeah,
Arthi.
yeah,
Uh,
yeah, yeah, yeah.
it was a good and that you explained all the technical terms here with Ramachandran plot, but
Yeah.
I also want to give a
Yeah, yeah, sure.
simple term like I just want to start it with a question: why everyone is using Ramachandran plot to validate the structure? Maybe everyone have this question in your mind. So the simple answer would be, let's take primary sequence, secondary structure, and tertiary structure. Like you can ask a question when you have a 20 amino acids with associated properties. When you put all the sequence in somewhere, it can form linear chain. Why it is folding like a why it is folding instead of linear? The answer is every amino acid has a particular properties. For example, some group of amino acids can form alpha helix, some can form turns, some form loops. So the Ramachandran plot already defined the limitation of rotation. For example, glycine is the small residue and proline has a closed ring. So these all the particular residues limited with particular dihedral angle rotation. So when you have a complete structure, in that case the Ramachandran plot will be used whether all the residues involved in the models are correctly rotated and whether it is in the allowed region or disallowed region. That's what we always need to go for Ramachandran plot to validate the structure. And this is one of the reason we kept this as a logo in LLB school as well. So this is the heart of structural mathematics. So yeah, Arthi, yeah, sorry to
Yeah, yeah, no problem, no
yeah,
problem, because they should know that.
yeah,
Yeah.
yeah. Thank you.
The next one is, so I have done all those things. I have modeled a protein, as Magesh said. I have done Ramachandran plot also. So is it enough to do and validate the protein? No, right? Because since we are doing computational method, you need to look for other criterias also. So there are n number of validation things are available for that score, and that score indicates how many standard deviation of an element here mention about the atoms which is present in the protein sequences, and to compare the results from a normal population. And error server also we can check and verify 3D. These things are available in a particular server called saves. Okay, SAVES. Okay, just note it on save servers. This is to validate your protein structure. Along with save servers, you can go for what if, and even I think uh,
What if
Magesh, what are
the classical
what
name
if
of saves is what if server?
yeah, water server, and we can go for saves or RCSP protein data bank and all in our upcoming classes. Okay, that will be helpful for you people. Okay, and in ERAT server you can check out the heavy atoms, that is your carbon, nitrogen, and oxygen. Okay, and you have 6 different combination, that is in, for example, you have carbon, nitrogen, and oxygen heavy atoms in your structure. You can go for carbon carbon, carbon nitrogen combination, and carbon oxygen combination, and nitrogen nitrogen combination, and oxygen oxygen combination. So you might be wondered how atoms are in your protein sequence, right? Then we will go for RCSB page and we will look for that. Okay, so how atoms are available, how the heavy atoms are present in our protein structure and all. Okay, and then ERAT will check for pairwise interaction. Okay, and then if it can find out the incorrect pattern of non-bonded interaction. In case if any of the combination is not available, then you will find incorrectly folded regions. Okay, and verify 3D, it also determines the compatibility of a 3D atomic level with the own amino acid sequence along with your structural environmental class, that is called your alpha and beta and loop and polar and non-polar thing. Okay, and distribution of polar and non-polar residues can be used to detect the error in the structure. Okay, I think Dr. Magesh has mentioned the link for RCSB.
Yeah.
Okay. Then this one is from Swiss PDB. I have mentioned here, no, there are a number of servers are available. This is an example for Swiss model. So based on the template, I got the first one is model, and the three model 1A, B will be available. Okay, and the third one is your template. Okay, so here it will be mentioned about alpha helix and beta pleated sheets and all. Okay, and even the template identity, here it is 67.97 97% identity. Not a good model, but you can consider it as a good model also. Okay, if it is above 80 or 90%, you can consider it as a model, consider it as a precise model. But here if it is 67.9%, you can again redo the homology modeling and you can have a better model. Okay, then superimpose I didn't mention here, probably I'll be mentioning in next class, and even you can find out the q mean also. So, and probably you have seen about chimera, right? So after you get a model, okay, you have your protein model, you need to go for Swiss PDB viewer or PyMol or SRR to view your model. Okay, there are, I have mentioned about commercial software also. Okay, and you can look for your model, and model refinement and evaluation can be done through verify 3D, ERAT plot, and Ramachandran plot. These things can be available in save server. Okay, so limitations and advantages and disadvantages. So what is the limitation? Accuracy, and the second thing is 50% sequence identity, which forms reliable models. Okay, and advantages: location of alpha carbons of key residues inside the folded protein, and you can which has been guided to mutagenic experiments also, and you can hypothesize this structure, and you can read about the function of the entire protein in detail. Okay, so for say for example, it is not that much easy to do crystal structure, right, in this current scenario for COVID-19. So you can go for this computational model and then you can refine the structure easily. Then the position of conserved region of the protein surface can help identifying active site. So probably what is active site, you know about the active site, right? Active site and binding pockets can be seen, and the how a drug is interacted, how a ligand can be interacted with a particular protein can be seen after homology modeling, that is called your molecular docking or molecular dynamic simulation. Okay, and the disadvantages belongs to this accuracy of the model, as I have mentioned in the limitation, the same thing. And drug designing and development process, little, you cannot say it's a disadvantage. instead of using a X-ray crystal structure, it is little difficult, but it's a good, however, it may be helpful for the same. If the sequence identity with the template is greater than 70, in the previous slide I have mentioned it is 67%. So if you take it for drug designing and development process, it is not, you will not get an accurate drug or accurate vaccine for that. But if you have 70% or above 80% identity, you can consider it as a good model. So with this, I'm ending the session. If you have any doubts, you can ask.
So thank you so much, Dr. Arthi. So guys, if you have any question, kindly let us know in the comment box, or you can unmute yourself and ask directly to Arthi. So any question, guys? Okay. I guess there are no.
Okay.
I guess there are no questions. So maybe we.
Yeah, in case if you find difficulty with any of the terms which is in the PPT, you can ping me or you can ask in the